Hello, my name is Christina Moore, and today I'll be presenting my plant disease portfolio covering two economically important plant pathogens, Rhizoctonia tursicum, which affects turf grass systems, and Phytophthora sojei, the causal agent of phytophthora root and stem rot in soybean. I will discuss the pathogen biology, disease cycles, symptoms, diagnosis, and management approaches for each. Zectonia tursicum is a soil-borne pathogen affecting both warm and cool season turf grasses, including bluegrasses and ryegrasses. The organism is classified as Mycelia sterilia, meaning it does not produce asexual spores. Instead, the fungus spreads and survives strictly as vegetative hyphae. One of the most important survival features is the production of sclerotia, which are hardened masses of compact fungal cells. These structures allow the pathogen to persist for long periods in soil and thatch, even under harsh environmental conditions. Rhizoctonia thrives in warm, humid environments, making the southern United States particularly susceptible. Symptoms of Rhizoctonia tursicum vary depending on mowing height. In high-cut turf, the first signs are circular brown patches, often with visible white mycelium early in the morning. In closely mowed turf, such as putting greens, we see what are known as smoke rings, thin brown or gray rings around the perimeter of the patch. Microscopically, hyphae can be identified by their characteristic right-angled branching with a septum located near the point of origin. These features help distinguish Rhizoctonia from other turf grass pathogens. This slide shows the disease cycle of Rhizoctonia. It survives in the soil or thatch as either mycelium or sclerotia. When warm and humid weather conditions occur, the fungus becomes active and begins to colonize turf grass surfaces. It first produces runner hyphae that grow along the leaf blade. The fungus then penetrates the host either through stomata or by forming infection cushions, which are dense clusters of hyphae specialized for invasion. Once established, the pathogen kills plant tissue, which results in the characteristic brown patches. Because the fungus is also an efficient saprophyte, it can persist in organic matter even without a living host, making management especially challenging. Managing this pathogen requires reducing moisture and improving air circulation. The pathogen relies heavily on high humidity, so cultural practices such as minimizing irrigation during humid periods and improving drainage are essential. Chemical control is also effective, especially preventative applications of fungicides such as carboxamides. These can be applied in fall when temperature drops below 70 degrees, which helps protect turf during high-risk periods. Combining reduced irrigation, proper fertilization, and timely fungicide use provides the strongest defense. Phytophthora root and stem rot of soybean is caused by Phytophthora sojei, an oomycete pathogen rather than a true fungus. Oomycetes belong to the Straminopola kingdom and share more characteristics with algae than fungi. This pathogen is highly host-specific and infects only soybeans. It survives in soil for over a decade as oospores, which are thick-walled and extremely durable. Under saturated soil conditions and temperatures above 50 degrees Fahrenheit, these oospores germinate and produce mobile zoospores that swim toward soybean roots. Symptoms depend on plant age. Young seedlings may be infected before emergence, leading to damping off. Those that do emerge often wilt rapidly and die. In older plants, symptoms begin as yellowing of lower leaves, progressing upward. Stem lesions form at the soil line and extend 2 to 3 nodes above it. As infection progresses, roots become soft, water-soaked, and eventually collapse. A key diagnostic sign late in the season is the presence of dark brown lesions extending up the stem while dead leaves remain attached. Pathogen's disease cycle begins when oospores germinate in waterlogged soils. They release zoospores, which are attracted to soybean root exudates through chemotaxis. The zoospores encyst, penetrate roots, and colonize the plant. Unlike Rhizoctonia, PSOJ is considered monocyclic, meaning it typically competes only one major disease cycle per season. However, environmental conditions determine the severity of infection. Extended periods of saturated soil greatly increase disease pressure. Because oospores can persist for more than 10 years, once a field is infected, it remains infected indefinitely. Diagnosis can begin in the field through visual symptoms such as stem lesions, wilted plants, and root rot. For laboratory confirmations, several methods are available. Microscopy can detect the characteristics of thick-walled oospores. Culture isolation on selective PARP agar helps distinguish PSOJ from other organisms. Molecular tools such as PCR or qPCR provide highly accurate identification, and ELISA tests can detect pathogen-specific antigens. Pathogenicity tests, where the pathogen is reintroduced to a healthy plant to reproduce symptoms, provide final confirmation. Management relies heavily on resistant soybean cultivars. Many contain RPS genes such as RPS1A, RPS1C, or RPS3A, which recognize specific virulence genes in PSOJ. However, the pathogen evolves quickly, so farmers often use cultivars with stacked RPS genes. Improving soil drainage is one of the most effective strategies because the pathogen depends on saturated soils. Seed treatments such as metalaxyl also provide protection during early growth stages. Economically, PSOJ is one of the most destructive soybean pathogens worldwide, causing losses of up to $5,000 per hectare and more than $2 billion globally. These impacts underline the importance of integrated disease management. References used in this presentation were taken directly from my 2 written essays.